we have studied that plasma membrane is selectively permeable membrane. That means it allows only some substances to enter inside the cell and then it allows few substances to go out of the cell. Now let us study that how this process of movement of substances in and out of the cell takes place. So first let us study about the gaseous exchange between the cell and its external environment. This gaseous exchange actually takes place by the process of diffusion. Diffusion is a process in which the substances move from a region of higher concentration to the region of their lower concentration, and the particles are very small in size, so they can easily diffuse through the plasma membrane. So movement of oxygen and carbon dioxide across the cell membrane is carried out by the means of diffusion only. In the diagram, we can see that the movement of oxygen is from higher concentration to the lower concentration. And again we can see in the second arrow that higher concentration of O2 is inside and then it is moving to a region of lower concentration. Further, the gaseous exchange substances can move from a region of higher concentration to a region where the concentration is low. Movement of water between the cell and its external environment takes place by the process of osmosis. Osmosis is the process in which there is movement of solvent particles from a region of higher concentration to a region of their lower concentration. But you have to remember one main point, that in the process of osmosis, a semi-permeable membrane is involved, whereas in diffusion there is no role of a semi-permeable membrane. So we also term osmosis as a special case of process of diffusion. So movement is through a semi-permeable membrane. This is the main point that we have to remember here. Let us study a little more about the movement of substances in and out of the cell. So first let us talk about hypotonic solutions. Now what is a hypotonic solution? In a hypotonic solution, student, the concentration of water outside the cell is higher than the concentration of water inside the cell. And as we have studied, the solvent particles will move from a region of higher concentration to a lower concentration. So in this condition, in case of a hypotonic solution, the movement of solvent particles will be from outside of the cell to the inside of the cell through plasma membrane. Now as a result, when water is entering the cell, as a result the cell gains water by the process of osmosis. Water can move into the cell from cell membrane, and in hypotonic solutions more amount of water enters the cell. When more amount of water enters the cell, then eventually the cell will swell up, and at times in this condition the cells can even burst. The bursting of RBC is termed as hemolyzed. Further, let us study about isotonic solutions. In isotonic solutions, the medium has exactly the same water concentration as the cell. So there is no net movement of water across the cell membrane. That means the amount of water that is entering the cell is same as the amount of water that is going out of the cell. So as a result, there is no net movement of water and the size of the cell will not change in this condition, that is isotonic solutions. Ringer solution is considered as an isotonic solution. Next, let us study about the hypertonic solution. Now in hypertonic solution, the medium has a lower concentration of water than the cell. Now the water will move out of the cell by the process of osmosis, as the outer concentration is less and inner concentration is more. So obviously the net movement of water will be from a higher concentration to a lower concentration, and the lower concentration is outside the cell. As a result, when the water moves out of the cell, it results in the decrease in size of the cells. And this condition in plant cells is known as plasmolyzed cells. And if we talk about in animal cells, then this condition is known as crenated condition.